Okay, cool. I think we'll just give it like let me turn off the waiting room actually now. So I think we'll give it like 1 minute and then we can start. Can you hear me?
Am I still not able to be? Am I still not able to be heard? Or
Anjali, I can hear you.
Okay. Okay. Good. Should we start? Okay. Hello, everyone. Welcome to the first UBIC Talk of the 2021 to 2022 school year. Today we have Dr. Bradley Voytek presenting his talk, Quantifying Neural Data, Neuroscience in the Data Science Age. Dr. Voytek is currently an associate professor with the Cognitive Science Department in the Neurosciences graduate program. He's also a co-founder of the Halicioglu Data Science Institute and UCSD's undergrad data science program. He obtained his PhD in neuroscience from UC Berkeley in 2014 and worked in industry as a data science before transitioning to university level research. Dr. Voytek's research focuses on using neural modeling, data mining, and machine learning to understand the mechanisms behind cognitive decline. And much of this research involves studying the relationship between the structure of the nervous system, the nature behind cognitive phenomena, and neural oscillatory activity. His lab has also created Brain Scanner and NeuroDSP, which are programs designed to computationally generate neuroscience hypotheses based on current research. Dr. Voytek is also the co-author of the book, Do Zombies Dream of Undead Sheep, a book about zombie brain research. He is passionate about speaking about science research to individuals of all ages and is involved in numerous science outreach programs. So please give a warm welcome to Dr. Voytek. And you can start whenever you're ready.
Thanks, that was great. Sorry, I'm a little sweaty. I'm coaching my son's baseball team. And so like, I almost forgot I was out there coaching and we're like doing batting practice and I got the alert on my phone and I was like, oh crap, I gotta run inside. So sorry about that, but I do have a presentation. Now, there's 12 of you on here. So it's small enough that I love giving a chalk talk. I know that normally the series, my understanding is that it's a chalk talk, right? Where I'm up on the whiteboard or blackboard. I love doing that. That's really hard to do over Zoom. So I have a presentation that I can give, but you've got me for the next hour-ish, right? And so I also wanted to use this time we can also have a conversation. People can unmute, they can chat about their interest in research or undergrad and grad school experience. Have any of you taken a class with me before?
I want to take neural data science next quarter. I'm looking forward to that class a lot.
Okay, cool. So you might be taking a class with me.
Yeah,
that's a new one. That's good. The reason I ask is because then the stuff I tell you won't be boringly repeating myself. As Anjali said in the fantastic introduction, I did work as a data scientist also in industry for a while. And so I've worked in the industry side and of course on the academic side. And the industry side, as Anjali also mentioned, my lab develops a lot of software. So we do a lot of Python programming, developing packages that other labs can use. And writing good code that is well documented that other people can use is one of the most impactful things I think I could do in my career. So on the data science side in industry, I was the first data scientist at Uber back when it was an 8-person startup. And so we were like a struggling, hungry startup, in a co-working space with a bunch of other startups. And, there were 10 drivers that like total in the world that drove with Uber. And we knew all of them by name and we hung out with them and we saw this company grow from 10 drivers to hundreds of thousands of drivers, and from one city, San Francisco, to just about every city in the world. And in industry and software development, software engineering, data science, that kind of stuff, they say that you can write code. The things that you do can impact thousands or millions of people, and that is true. But when I was working at Uber, the code that I was writing would maybe get, an amazing day would be, or month or project year, would be if you wrote code that got people a car a couple of seconds faster on average, right? That would be an amazing feat to do, where you figure out some algorithm that makes it so on average, cars get to people a little bit quicker, right? Because then for the company, that means that They can do more trips per day and make more money, and the drivers can earn more money, and everybody's happier because all of you, if you've ever taken Uber or Lyft or whatever ride sharing service, you get a car a little bit faster, it makes everybody happy, which is an amazing thing to be able to do, where you write code that touches the lives of that many people potentially. But on the, is this fulfilling side for me, that wasn't scratching that itch. I did my PhD in neuroscience. I did my undergraduate in physics initially. My degree didn't end up being in physics. I started as a physicist and was terrible at it. And I'm not saying like, oh no, I got a B sometimes. I mean, like actually terrible. When I went to register for classes in my third year, I was told I couldn't because I'd been on academic probation for too long. So I was kicked out of the university. That level of terrible. And I had to like beg and plead with the undergraduate academic advisor at my university to explain, I mean, there were reasons other than just me not studying, like there were reasons in my life, my personal life, what was going on that made my academic journey at the beginning in undergraduate difficult. And I explained those to the academic advisor and sort of showed the receipts. And she gave me a second chance. If this one person hadn't really gone to bat for me and was like, oh, okay, let's try and give you a second chance, my life would be very different. And so I also have that experience as an undergraduate. I know that UC San Diego, especially in biology, is considered to be a very competitive environment sometimes. As a faculty member, that hurts me, right? We want you to be collaborating together on things and working together, but I understand it's also very competitive. And that is stressful. And so I'm happy to also have that conversation too, if you would like. But anyway, when I write code in industry to touch people's lives, that's great, but it wasn't, like I said, scratching that itch for me to know that, initially I thought, well, maybe I'll go to medical school. Because I know that in medical school, if I get an MD, I can see patients and I can improve the quality of people's lives directly, right? Ideally, you could be saving people's lives when you're working with individual patients. But, if you're a medical doctor and your career starts in your early 30s and you do it for until you're 70, right? You've got 40 years career where you can work with patients. And how many people can you see on a one-on-one basis, right? You have the potential to help thousands of people. But if I go into research and I write code that makes academic research easier for other labs, thousands of different labs around the world, maybe I can actually have the potential to, in the same way in industry, where you can write code that touches thousands of lives, maybe I can write code that improves research that could help potentially millions of people. I'm not saying that's going to happen. It may very well be that I have a 40-year-long academic career running my research lab and we write a bunch of code, but it doesn't help anybody ever. But that's part of the scientific enterprise, right? Is that maybe what I do right now or in my entire career doesn't directly help, but we're building that edifice up so that other labs can then build upon what we have done and make things better, right? And so to me, in thinking about what do I want my life to be, I could be making a boatload of money, and I walked away from a boatload of money at Uber because we didn't see eye to eye on lots of things, the early leadership of Uber and I, versus running a neuroscience research lab where you could do something that maybe help people with Alzheimer's disease, dementia, schizophrenia, autism, who knows, right? These are diseases, disorders that impact millions of lives around the world and will continue to do so going forward, decades into the future, barring the identification of any cures, right? Aging is something that influences our cognition and cognitive performance. And maybe we could figure out something that slows that cognitive decline down as we get older. I mean, maybe that's a little bit self-serving. Also, as I am now getting more gray bearded and older, but so this is what motivates my lab, is this idea that we can write code and think about the brain in ways that might make other research better. And that's it. That's like the foundation of what we do. And in like super ridiculously simplified terms, what my lab actually does is we take huge amounts of neural data that people are collecting, and ourselves included, and we turn it into numbers better. That's it, right? You can't fix what you can't measure. as DJ Patel used to say. DJ Patel, by the way, is arguably the person that coined the phrase data scientist. He's a UCSD alumnus. He was the first chief data scientist of the United States under the Obama administration. So he has amazing stories of his days in the White House working with Obama as the first chief data scientist. And he has this great phrase, which is, you can't fix what you can't measure. And that's what we do. We measure brain activity, and my lab says, how do we turn that brain activity into numbers that make sense and that we can do better math on? And in order to make that happen, we have to write good code. And so I could go into all the details of what that looks like. I can start my presentation, but I wanted to give that preamble for this 8 to 10 minutes here, just to sort of set the groundwork and say, I can pause and go into the details of that. We call it the neural data parameterization and how we use data science on the neuroscience side. But I also wanted to pause and open it up for any questions that people might have, because it's a small enough group, we can do that. And I teach very large classes, and so I miss being able to have closer one-on-one conversations with students like this.
I actually had a question.
Yeah.
I was wondering how your PhD in neuroscience specifically helped you in industry and Uber specifically. Those are kind of you wouldn't think there's a lot of intersection in those two.
Well, so surprisingly, perhaps, lots of people that get PhDs in neuroscience go on to do data science. So if you do a PhD in neuroscience and you're in a wet lab doing the more neurobiology side, then your industry options are a little bit different. Oftentimes you're not doing large scale data analysis, you're doing things, you'll end up working for pharmaceutical companies, Genentech, these big companies, right? That's a common path, non-academic research path that people with neuroscience PhDs go into. Now, the other side, though, is the more mathematical computational neurosciences side. These are people that, you get terabytes sometimes of data from recordings from either human brain activity or animals, and learning how to write code to algorithmically go through, clean those data, and do relatively sophisticated analyses on those data sets. That lends itself pretty nicely to data science. Now, in my particular instance, it's a pretty wild story, which is one of my best friends from high school. I was the best man in his wedding. He and I had weird homes. He was, single mother, and I had my own weird situation. And so he and I used to just kind of hang out a lot as high school students. And this is in the mid to late 90s. And programming and hacking was a thing that people were excited about, right? It was coming out. And so he was pretty early on that side. He dropped out of undergraduate to do the sort of late 90s .com boom startup life, while I went on to do grad school. And I was supposed to, after I got my PhD, I had a job offer in Germany at Max Planck Research Institute to continue my research. And my wife and I went out there to visit, and we're both from California. And we're we can't move to Germany. We can't. It's so far from family. Everything is gray. There's weather. It's cold. And we're from Southern California. We don't want to do this. And so I finished my PhD. I had a job offer. I turned that job offer down. And now I'm 30 years old. And I'm I don't have a job. What the hell am I doing with my life? I've got a PhD and I'm not doing anything. And I was complaining to my friends about this, one of whom is this guy, Curtis Chambers, who was my buddy from high school, who dropped out to do the startup thing. He ended up in the Bay Area, because that's where startups were, doing their thing in the 2000s, where I was doing my PhD also. And I'm complaining to him, and he's well, hey, look, I just started working for this little startup, and you did data analysis in your PhD, right? Why don't you come talk to us? And so he was employee number 4 at Uber, and he's the one that brought me in just through total random chance. But that's an unusual circumstance. That said, lots of people in neuroscience do go on to do data science because there is a lot of overlap. The trick is recognizing that overlap exists. If I have a long time series of neural data, I've stuck an electrode into an animal's brain, or recording EEG or fMRI from somebody, or recording single neuron activity from a Plizia sea slug, or doing whole brain recordings from a zebrafish. It doesn't matter. These are all sampled data points over time. And the math that you do to analyze those data is just the same as trying to figure out how do people move around a city. Like, how often do people come and request a car, at a ride sharing service, and from where? The math is all the same. Linear algebra, time series analysis, all this kind of stuff. So the foundations are there. It's just sort of flipping that switch in your brain and thinking about, oh, rather than neural activity, I just think about how do people move around cities, and rather than how do neural signals move around, move between neurons. I know it sounds weird, but it's a really great, computational neuroscience is a fantastic set of skills that is broadly applicable to many research domains, and really cool careers in data science and software development and stuff like that. All right, so how do we turn data into numbers? Sounds stupid, right? I'll go ahead and start, I guess. Share screen. Oh, you got to let me share screen. I think it's probably in Jolly. Or is it Aditya? Okay.
I think Julia has to do. Oh. Yeah.
And then say share screen. Right there. Yeah.
Okay, there you go. You should be able to share screen now.
There we go. And let me do the standard settings, optimize for video, and let me get my pointer. Y'all see the spotlight there? Cool. Oh, you know what? Let me also open up the chat window in case. Feel free to also chat in here. Teaching during COVID made me get pretty decent at being able to monitor that while also talking. Okay, so this is my research lab. Actually, a little bit different right now. Natalie Schwarnikov, my postdoc, just moved on to a research career in Germany. So I have a full-time software developer in my lab, Ryan Hammonds, several neurosciences PhD students, Andrew Bender, MJ Preston, Sydney Smith, Blanca Burgos Martin, and Trevor McPherson, and 2 CogSci PhD students, Ina Kosick and Kirina Van Ingen. Probably going to be taking data science PhD students. So the Lee Jolu Data Science Institute is launching a PhD program starting next fall. So we'll be taking applicants this fall for neuroscience or data science PhDs as well. So this is the research crew in my lab right now. And, being in San Diego, I always have to throw the San Diego pick out. And during the pandemic, this is, we played a lot of Among Us and stuff, trying to continue socializing as a group. And everybody thought it was hilarious every time they got a chance to murder me. And the research that we do sits at this intersection of neuroscience. We have funding from the NSF to do neuroscience and data science work, and from the NIH to do software engineering. Right? So we really do all these kinds of different approaches. And when I started my lab in 2014, I started with this idea that oscillations are neat, neural oscillations. So I don't know if anybody's taken an intro to psych class or cog-sci. They might talk about it a little bit in neurobiology, might have heard of neural oscillations, which is if you stick an electrode into anything's brain, sometimes you see these rhythmic repeating semi-sinusoidal patterns. Right? Think of a sine wave, kind of see that sometimes. First signal ever recorded from a human brain in neuroimaging in the 1920s by Hans Berger, who discovered the EEG, electroencephalography, where you stick electrodes on people's heads and you could record brain activity. Noticed a very strong, clear oscillation that cycles at about 10 cycles per second over the visual parts of the brain, called it the alpha wave. And I was like, this is really cool. There's tons of people studying this. It's almost a 100-year-old signal in the sense of when we first discovered it. We still don't know if it's functional. Is it doing something in the brain? There's lots of really interesting theories and computational models that synchronized oscillations between 2 different brain regions maybe facilitates the ability for those brain regions to communicate with one another. Now, there's really strong physiological research going on the oscillation side, but brain waves entered sort of the public consciousness in the 1950s and 1960s, and now there's like this crazy field of oscillations that has run away from science entirely. So there's a problem with oscillations, which is on the actual science side, oscillations are correlated with everything. Neuroscience is a pretty old field. In fact, if you go to PubMed, if you've ever tried to engage with the peer-reviewed scientific process, PubMed is run by the NIH, and it's a repository of all the peer-reviewed science that's been published in biomedicine, at least. And there are approximately 3 million peer-reviewed papers in neuroscience. So if you want to be a neuroscientist, that's the backlog of peer-reviewed science you have to catch up on. Good luck. And in those 3 million papers, there's lots of stuff published. And so the way that we talk about brainwaves is they have different names. I already mentioned alpha, which is about 10 hertz in the visual cortex. There are beta waves, which are approximately 20 to 30 hertz, usually in the sensory motor parts of the brain. There are delta waves, which are slower and usually around 0 to 4 hertz. And those get really big when you're unconscious and like anesthetized. There are theta waves, which are 4 to 8 hertz. Those are very strong in the hippocampus, right, the memory part of the brain. And there are gamma rhythms, which are faster, usually around 40 to 80 hertz and so on. So you've got all these Greek letter names for these different oscillations. If you go into PubMed and search gamma oscillations and whatever, pick your disease of choice, pick your cognitive behavior of choice, language, memory, attention, whatever, you'll find papers talking about that oscillation, alpha, beta, gamma, delta, theta, and whatever you care about. Somebody's looked at the link between those and found it. And so they're just correlated with everything. And I mean everything. In fact, Francis Crick, a Nobel Prize winner with pretty deep UC San Diego roots, and Christoph Koch, who is now the Chief Science Officer at the Allen Institute in Seattle, they published this paper in 1990, which is, can't believe that 30 years ago now, talking about that the neurobiological basis of consciousness is probably from the coherent semi-synchronous oscillations in the gamma 40 to 70 hertz range. They had a theory. They came up with this idea that maybe consciousness arises by the coupling of these oscillations in the human brain. It's a theory paper. They're musing, came up with this idea. Now, if you go to the strange depths of the internet, you will find that this 40 hertz is the key to consciousness. And if you want to go down the crazy rabbit hole that is the internet, search gamma 40 hertz oscillations in consciousness, and you'll find people that are biohacking themselves with psilocybin and ketamine and DMT and binaural beats and meditation to increase their 40 hertz gamma oscillations to a higher state of consciousness, right? So there's tens of thousands of these papers talking about this stuff. And what my lab is stuck on is, what the hell do you mean when you say an oscillation? So first of all, I'm not sure this is good advice, but I would strongly recommend befriending philosophers. So pretty much the entire basis of the success of my lab has come from informal conversations that I've had with a very dear friend of mine who's a philosopher, where I talk about my research and he says, "What do you mean by that?" I'm like, "Come on, man, I'm not going to teach you the math." He's like, "No, what do you mean by an oscillation?" I don't know. I was like, "Well, it's like a rhythmic thing. Like, how do you know if you have a rhythm in like noisy neural data?" "Well, come on. Why are you asking me? Don't be stupid." And eventually, if you talk to a philosopher long enough, they break you down and convince you that you don't know anything about anything. And sometimes that's fruitful, because it turns out that if you push it far enough, it turns out maybe the field doesn't know what they're talking about, which is where the success in my lab has come from, is what are we talking about when we say an oscillation? If you're going to talk about neural oscillations as being critical for this disease or cognitive function or whatever, you got to be really clear about what you mean when you say an oscillation. So now in 2021, this is where my lab is stuck. We don't even know what we're talking about anymore. We don't even know what an oscillation means. We're pretty convinced that the field doesn't know what they mean. And we've done a lot of research the last couple of years trying to prove that, which is a very weird place to find myself, running a lab trying to turn around to this field that has been decades long and say, actually, we don't think y'all talking about what you think you're talking about. And then here's the proof, which is actually, I got to admit, pretty fun. But anyway, so this is the foundation of the lab. We can't talk about oscillations as anything meaningful scientifically without first understanding where do they come from. And second, without understanding how people traditionally measure oscillations. And now this is where you start getting into the weeds of the math and stuff like this. So I'm going to try and go a little bit slower and make sure that people are all on board. So one, where do oscillations come from? when we stick an electrode into something's brain, unless we're right into the neuron, so you stick your recording electrode, penetrate into the neuron itself. That gives you an intracellular recording of that neuron. That's where you get this bottom trace of action potentials, right? Otherwise, if you don't penetrate a single neuron, it's pretty hard to do, by the way. Most of the recordings that people do are extracellular, outside of those individual neurons. And those extracellular recordings are not picking up and just a quick reminder, action potentials are the output signals of the neurons, right? If you've taken a neurobiology class, can somebody define an action potential for me? What is it? Give me the great answer. Come on, you're neurobiology students in here. What's an action potential?
And it's a change in charge across the gradient, basically the neuron firing.
And what's the phrase that people like to use about an action potential? Do you remember? I know it's kind of vague. Electrochemical
fire
signal.
together, wire together.
Yeah, heavy, and that's heavy in learning. Neurons that fire together, wire together. But neuron action potentials, depolarization, that's the technical term. It's usually referred to as the all or nothing response, right? You either get an action potential or you do not. There is nowhere in between. So the history of AI and deep learning also has deep roots at UC San Diego. So in the 1970s and 1980s, there were a group of nerds at UC San Diego that were all about artificial neural networks, which was super boring, nerdy stuff that nobody cared about in the 80s, right? I should say very few people cared about. There was a crew of people at UC San Diego that worked at the intersection of neurobiology, psychology, and computer science, studying. They're like, oh, wait, maybe the all or nothing response of neurons, these action potentials, they call them binary. It's either you have an action potential or you do not. It's a 0 or a 1. It's kind of like how we do code in computers, right? It's, we've got zeros and ones. That's it. That's the binary code. Sorry. I don't know why I have a home phone. This isn't 1960. Anyway, so it's either all or nothing. And these people are like, well, maybe there is something we can learn about, how the brain works by developing code to simulate it using binary communication. And maybe we can learn something from the brain that will inform, we can encode it and maybe like create artificial intelligence. And so that group of people founded the cognitive science department here. One of the first people in the cog sci department was Jeff Hinton, which if you are a deep learning person, you will know that name because arguably one of the 3 co-creators of modern deep learning. And that grew out of this conversation between the neurobiology people and the computer scientists. That's where cog sci came from at UC San Diego. Now, these neuron action potentials are considered to be all or nothing. And one thing that my lab has been pushing at recently is that that's probably not true. Actually, you look at this trace here. Do you see how this first action potential is a little bit taller than that second one? And then it's, it goes back up and then goes back down. Do you see there's like a little bit of variability also in that resting membrane potential? Oftentimes, neural spikes are quantified as 0s or 1. It either spikes or does not. And that's how most people analyze spiking data. My lab has been writing code to take each individual action potential and precisely measure how tall that action potential is, how deep this dip afterward here is. Sometimes it exists, sometimes it doesn't. There's a little bit of variability in the action potential heights. And it turns out it's looking like all of that little tiny bits of variability is actually meaningful in carrying information. And we wouldn't know that unless you were able to write code and do a little bit better. Could those smaller variations be leaking ions across the gradient? Totally, right? The point of my lab isn't, we're not a wet lab. We can't do this. We don't know. I mean, we steal data from other people, not steal. We take data from other people all the time, right? And we really dig into it to try and figure it out because of hypotheses like that we can test. But our point is, you've just come up with a hypothesis about where that variability in action potential amplitude might come from. We can't test that hypothesis unless we precisely measure that variability. If you do what most people do, which is turn this really rich set of information here in this spike train into zeros and ones, is there an action potential or not? You've lost that variability. You've lost that ability to interrogate interesting physiological questions like you just posed, Aditya. And that's what we are, that's what my lab does, is we say, wait a minute. The way that we analyze these data of taking this rich signal and resampling it to zeros and ones is done for historical reasons of computation was expensive and hard drive space was expensive. And so people would record these action potentials, and they historically would have code in real time to detect, did the membrane potential go above a certain amplitude? If it did, mark the time of when that happened. So in order to get an action potential, they sample each electrode, they sample 20,000 samples per second, 20 to 40,000 samples per second, right? That's a lot of hard drive space, because to quantify the amplitude 20,000 times per second requires a lot of hard drive space, a lot of storage. And older computers didn't have enough storage to record very long studies. And so they would simplify their data analyses by just recording the time of when a spike happened. So you take this huge amount of data, 20,000 samples per second, and you reduce it to when did an actual potential occur, and that's it. Saves in hard drive space, makes data analysis a lot easy, but we're in a place in the world now, computationally, mathematically, storage-wise, where we don't have to keep doing that. Let's look at the richness of our data, right? Okay, so how are noise peaks usually separated from actual ones? So the way that most labs identify when an action potential occurs, the common approach is to just set a threshold, right? You look at this and you can see the noise levels down here, right? And so you just pick a voltage value and you say, well, the noise rarely, if ever, crosses like here, right? Whatever this line is, you pick a threshold. So that's it. That's how people do it. They pick a threshold and they just say if it rises above that threshold, then count it as an action potential. Is there any idea of the richness of that signal? Is it analog, or is there some kind of digital component to it? I mean, the real world is analog, right? The brain is analog. And we digitize it to make data analysis possible. Now there gets a little bit more complicated. There may be digital components. Maybe neurons' action potentials really all or nothing binary signals. If that's the case, great. Neuroscience becomes way easier. I mean, not easy, but easier if it turns out that individual action potentials really truly are binary all or nothing signals. I do not think that's probably the case, partly because these all or nothing action potentials are they're the majority, but they're not the only kind of signaling that occurs in the brain. There's a whole class of neurons that have graded action potentials, where not even action potentials, graded signaling, where it's not an action potential all or nothing. It's just a continuous gradient of voltage values, and the neurons are physically connected with each other rather than by synapses, which are the gaps between cells. Some neurons are physically connected to each other. They share, it's called a gap junction. They share transmembrane ion channels, and they actually pass ions directly between them in a graded fashion. One of the core animals, anybody work as an undergraduate in a neurobiology lab? Do you do research? Anybody? You do? Have you worked, what species do you work with? What's your like model organism in the lab that you do?
I do mostly like microimaging analysis using.
Somebody who works with mice? What's okay, microimaging analysis, and what species? Marmosets. Oh, okay. Are you with Corey?
No, I work at Salk under Uri Manor and with like.
Oh, okay. Okay, there's a classic, nobody's really saying it. There's a classic model organism. Have you guys heard of C. elegans? Have you heard of this? It's a nematode. C. elegans are pretty awesome, right? Because they're only, Michelle is on here. I don't remember, is it 1,000 something neurons? What is it, 1,021, right? It's the only animal where we have pretty much every neuron identified as to when it develops and how it's connected. Now, here's the crazy thing about C. elegans. They don't actually have action potentials. So this like model organism that we use to understand neurobiology, and we have every neuron is like named. We know when it arises during the development of the little tiny worm, and what it's connected to. But they actually don't have action potentials. It's all just these graded potentials. So anyway, these are the kinds of things that we're interested in, is looking at this variability, leveraging the rich variability of the signals we have rather than treating most of it as noise. And so you can record intracellular active potentials or extracellular, which is this local field potential, LFP. And the intracellular recordings of the active potentials are the outputs of the neurons. When you have extracellular recordings, what you're largely picking up are synaptic and transmembrane currents. So that means not the outputs of active potentials, but the inputs to that region. So the currents that are being induced postsynaptically by wherever those inputs are coming from, where the ion channels are opening and closing, and these are creating different currents. So you begin with a basic physiological understanding of the signals you're recording from. And we know that this is the primary origin of the extracellular recordings that we're picking up. And so, beginning from that, we know that these postsynaptic and transmembrane currents have what we say here in the upper right. In the time domain, where you look at the time on the x-axis and an idealized synaptic kernel here with voltage on the y-axis, is characterized by its double exponential form, meaning that the voltage exponentially rises and then exponentially decays again. But the exponential rise is faster than the exponential decay and has this characteristic double exponential form. And so this is all just computational modeling, none of this is real, it's all simulated data. On the left, you have a pretend neuron receiving many inputs from all of these different regions, and they're getting all of these little postsynaptic currents that are being induced at different times in the neuron. Those sum up to this extracellular local field potential. And in the bottom right is the same signal as the top, which is the time domain signal, but this is what's called the power spectrum or power spectral density of this signal, the PSD, which is rather than voltage by time, your signal can be perfectly mathematically represented as a sum of sine waves of here the y-axis is the power or amplitude of those individual sine waves by frequency here on the x-axis. So a sine wave of 1 hertz, 2 hertz, 3 hertz, 4 hertz, 5 hertz, 6 hertz, 7 hertz, all the way up to 150 hertz here or so. And you add a bunch of sine waves together and you get this signal here. This is what's called the Fourier transform. It's freaking math magic. It's amazing. Anything that can be represented as a regularly sampled signal in time can be perfectly mathematically represented without any loss of information as a sum of sine waves of different amplitudes and frequencies. What happens is these synaptic kernels have different shapes in the time domain. So this would be a synaptic kernel that might be inhibitory. It has what's called a long time constant here. It takes about 200 milliseconds to rise and then start to decay back down by 300 milliseconds. It's pretty much back to baseline. Whereas an excitatory current is faster, rises quicker. And so we're stepping between inhibitory and an excitatory current and simulating what this looks like and what the LFP looks like and what the power spectrum looks like. Because the reason I'm showing this is how people define oscillations. They take their signal and they break it up into these different frequency bands using this Fourier transform. And they say, look, we have delta is defined as 1 to 4 hertz. And so they'll take the power in this 1, 2, 3, 4 hertz range here, and they'll average it, and that's the delta power. That's the power in the delta rhythm. Alpha power is like 8 to 12 hertz. And so we're going to go like in this range right here. 12 is, or log axis here, right? So 12 is like here, right? So power in this range. But the reason I'm showing this to you is by definition, this signal that we've created mathematically or in simulation has no oscillation in it by definition. But because we know and understand the physiological origin of the EEG signal, the local field potential, these extracellular recordings, they will always have power at every frequency due to the mathematical nature. And I'm not going to cover like the exact proof as to why this is, but anything that is represented by a double exponential in the time domain has this characteristic. It's called the Lorentzian in the frequency domain. We have relatively equal power out to some what's called the knee frequency, after which it bends down and then becomes linear above a certain frequency. And so the reason that I show this is most of the data that we have looks more like this in the bottom right. Most of the neural data doesn't have an oscillation in it. It just has this shape, which is, we call it aperiodic as opposed to periodic. So periodic meaning oscillatory. Most neural data is aperiodic. And this is a signal, this aperiodic feature that people have considered to be noise. And what we're arguing is that this aperiodic signal is signal, not noise, if you understand the physiology of what's being generated. This aperiodic signal can change. So you notice if I have a signal purely driven by excitation here, like in purple or pink now, versus going back towards purely inhibition, this power spectrum in the bottom right shifts around. It's dynamic, it's modulated. And that's important because here, if I stop and I have like two groups, let's say, of people, one with a disease and one without, and let me move my controller here so I can hit pause. And I say, I think, my hypothesis is that alpha power, 8 to 12 hertz, and like right here, is characteristically different between group A and group B. Here, if I have one group that's dominated by excitation and another group that's dominated by inhibition, and I look at the power at 10 hertz, they're markedly different, right? So if I analyze my data by looking at the power at 10 hertz, this alpha range, as people traditionally do, I will find reliable, replicatable group differences that say alpha power is different between group A and group B, or cognitive condition A and rest or something like that. But it's wrong to say it's an oscillation. And so this is what my lab does. What do we mean by an oscillation? So I study neural data. This is an example of neural data here. And I can look at my neural data here and I say, well, I'm used to looking at this kind of stuff. Where's the oscillation? Maybe here it's cycling for a couple of cycles. Maybe that's an oscillation. It's going for more cycles. Maybe there's like a really fast oscillation here. That's a lower amplitude. How do we know what an oscillation is? Does the pattern of the curve change depending on the region of the brain for which you're trying to obtain the signal from? Yes, it does. And I'll show you some results about that. It's actually, there's a smooth gradient across the human neocortex and macaques for the pattern of that curve that we argue tells us something about the computational properties of the different parts of the cortex. So I'm going to skip over this. We have a great collaboration with Alison Motry here at UCSD using cortical organoids because we can grow cortical organoids. I'm not a biologist. In fact, I haven't taken a biology class since high school. So, I pretend to be a biologist. So I'm just going to wave my hands here and say, through the magic of cell biology that I don't understand, apparently we can take skin cells from people's cheeks, like a cheek swab, and then we can trick those into becoming pluripotent stem cells and then grow them as brains. Apparently we have that ability. And so this is a collaboration with Alison Motry. They take those pluripotent stem cells, they grow them forward as neural tissue, and then we query those, we record electrical activity from these cortical organoids. They're about the size of a bee's brain. They're about 100,000 neurons. And we watch them over development to look at when do oscillations develop. And it turns out you actually see real oscillations, like here's a signal oscillating in time. You can see it spontaneously emerge through just normal genetic pre-programming of how excitatory and inhibitory neurons seem to develop and wire together. And those oscillations spontaneously begin to emerge after several months and then begin to differentiate into different frequency bands. And so we have a pretty cool collaboration that's ongoing with Alison Motry's lab. They do the cell biology magic on the organoid side, and then we do the computational modeling and data analysis of them to try and understand physiologically where do oscillations come from. At what point in this development did they arise and what changed in that development? Was it the wiring between excitation and inhibition? Was it early in development, GABA, which is the primary inhibitory neurotransmitter, is actually excitatory, and then it flips sign and becomes inhibitory later in development? Everything is hyperconnected synaptically in early development, and then those synapses get pruned away. And so, like, what is the actual physiological basis? What has changed between 4 months and 6 months? At which point you see no oscillations or pseudo oscillations to very clear strong oscillations. So that's step one of what we were talking about earlier, is we can physiologically interrogate what gives rise to these oscillations. Where do they come from? And step 2 is how are they measured, right? And so I'm going to end here in the last couple minutes, just so we can have more time to chat. So this is the crash course now in the Fourier transform. Anything, like I said, that can be represented as regularly sampled over time, here voltage on the x-axis, time on the y-axis, is perfectly mathematically represented as some of different sine waves of different frequencies here on the x-axis, and voltage or amplitude, also power on the y-axis. And you can see this signal here is composed of 3 different sine waves, mostly of the same amplitude, and then a bunch of really low level noise. Neural data, the frequency domain looks like this. So remember that knee and that curve that I showed you before? That's what real neural data looks like. We argue because we understand that it's being driven by this thing, right? It's like a great mystery that we seem to have helped people figure out. But then you see these bumps. Do you see this bump right here, like 8 hertz? What is that, 10, 9, 8? Yeah, like 6 to 9 hertz maybe. You see have this like little bump here where this is logarithmic on the y-axis, so it's exponentially higher power than this sort of background curve. And there's another little sub bump here, right? That is an oscillation. There is a narrow frequency band where there's extra power above and beyond whatever this like background knee is, right? And that's how an oscillation is defined. Here's the Fourier transform in action. You can actually see it. So you have this black signal down here. And as you add more and more sine waves together here on the top, you get the red signal on the bottom, which is a better, closer and closer approximation of the raw data. And so each sine wave is characterized by, down here, it's power again on this axis. Unlabeled axes are terrible, but it's a GIF, forgive me. Frequency on the x-axis, and then the phase, the starting phase of that. Like, does it start at this phase or more toward like the peak or whatever? So I was just showing the coolness of the Fourier transform at work. And so to go to Michelle's question, I'm going to skip ahead and stop sharing and just show you that there's really neat genetic patterning across the cortex. Do I have it? Yeah, there it is. This is the neural data science side of what my lab does. So we had this hypothesis about what this shape means. So remember the power spectrum I said is flat out to a certain frequency, and then it hits that knee frequency and bends down. And so we argued that the shape of that tells us something about how correlated a time series is with itself. I'm not going to do the mathematical derivation for you. But signals that are more auto-correlated mean that if you look at any time segment of the signal, it looks more like itself longer in time. So signal is correlated with itself in time. You can think about it roughly as like the memory. If a part of the brain gets an input, how long does that signal maintain before it decays away? And that's called the autocorrelation of the signal, which is looks like this knee sort of shape in the frequency domain. This is the time domain, frequency domain, and we had this hypothesis about what that might mean about memory. And so we took a bunch of open data sets, intracranial recordings. My lab works with people who are undergoing brain surgery. So we can record brain activity directly under the skull from them, because the skull is neuroscientifically very annoying. It gets in the way of our ability to record signals. I mean, like, I'm glad I've got one. I'm glad you've got one. Hopefully you've got one. But from a signal processing standpoint, it's very annoying because it gets in the way of all of our signals. And so we work with people who are undergoing brain surgery for epilepsy, and they're monitored for 2 weeks at the epilepsy monitoring unit. The surgeon that we work with here is Sharona Ben-Haim. She's the brain surgeon, and she implants electrodes. This is done all around the world for decades, directly into their brains to monitor what part of the brain is causing seizures, the epilepsy. During that 2 weeks when they're being monitored, they've got electrodes implanted in their brain. They're hanging out at the epilepsy monitoring unit, watching TV and playing video games and reading books and stuff, waiting to have seizures. During that time, my lab approaches them and asks them to take part in our research because they offer an unparalleled access to brain activity in humans that you can't otherwise get in any way, right? And we took a massive data set of these intracranial recordings collected from a bunch of collaborators over many different sites over many, many years. And we integrated that with structural brain mapping. Let me see if I can get my pointer back. There we go. Very close, very close. There we go. Oh, no, I lost it. Shoot. Yeah, there it is. So we integrated this data set of huge amounts of intracranial recordings from people with structural brain scans from several thousand other people to look at average shape of the cortex and how they're interconnected in different brain regions. And then another set of data from the Allen Brain Institute in Seattle, which has gene expression from 6 human brains post-mortem for the 20,000 or so different genes that are differentially expressed. And we integrated all of those different data sets to see how does this memory activity from the computationally derived time scale, the thing that I was talking about earlier, from the intracranial data relate to structural gradients in the brain and transcriptomic gradients in how different genes are expressed. And we found really cool correlations and relationships between the smooth sort of patterning of these timescales, which are very short and rapid, meaning in like visual and sensory regions, and motor cortex, meaning the regions have very low memory. They get an input and they quickly forget it, whereas frontal cortex and hippocampus have much longer memory. The signals look more similar over time. So they get an input and they maintain that input for longer and longer timescales. And that's correlated with the degree of myelination anatomically across the cortex, as well as genes that encode for excitation and inhibition. But anyway, that's too much. I'm going into way too much detail, so I'm going to stop because you're all probably lost, and that's no fun. So let's just chat. Neuroscience is freaking cool, you all. Like, there's so much to do. We have so much data out there, and we understand so little about how brains work, right? I mean, it's really an amazingly fun job just because there's so much opportunity. This is not how I started. When I applied to grad school, my application, my research statement for my PhD was to look at whether video game addiction had the same neural basis as drug addiction. That was what I was thinking I was going to do for my PhD. This is nothing like that, right? And so a huge part of research is just kind of going in interesting directions. Don't set yourself down on a specific path and say, now I'm stuck doing this, right? I guess I'm an addiction researcher. You know, you have some flexibility and opportunity. You just have to take advantage of the resources that a large university like UCSD has, right? Take advantage of the office hours of your professors and TAs, talk to the grad students that are doing the research, talk to postdocs that are doing the research. Most faculty members, you know, I hate to say it, if you email them, they're probably going to ignore you because we get deluged by emails. Not all of them, you know, I try to answer emails, but some of them will ignore you. You know, brush it off and continue on and, you know, take advantage of the research opportunities, the staff that work here, the student advising staff and stuff like that. Really like get to know people. You've got 4 years. I know it seems like a long time, but it goes by like that. And so you should take advantage of every opportunity that you have here, especially now that you're back in person, right? Like carpe diem, you don't have a lot of time anymore, right? Like we could have a pandemic again next year and stuck at home. God, I hope we don't have a pandemic next year. I'm just saying it could happen, right? Like, and so, you know, run with the time you've got.
I actually have a question about grad school. So I'm right now a third year and I want to apply in computational neuroscience, but not every university has like a computational neuroscience program. So I'm kind of wondering like how to pick a program, because I could see myself doing like CS, cognitive science, data science, math, neurobiology, etc. And I just don't know, because obviously I can't apply for all of them because of money and letters of rec and whatnot. I'm just kind of wondering like how do you choose what program to apply to?
Well, money may not be a problem. If you actually have financial hardship, most universities will waive the grad application fee. And so what you need to do, and this is advice for everybody, is for every program that you're considering applying to, go to the website, because almost inevitably every university will have their website, and they will list the staff that work for there. Look for the person whose title is something like graduate coordinator, usually is the title. It's a full-time staff member, it's not a professor. They'll answer your email and let them know that you want to apply to the program, but you have a financial hardship. And they will almost certainly always waive that application fee. The other thing is if you apply to UCSD, Michelle, maybe you can correct me because you applied here, but if you apply for one program, you can apply for multiple programs at the same university, usually without incurring another fee. And finally, regarding letters of recommendation, a lot of programs now have letter writing services. So if you ask me for a letter of recommendation, I write one letter for you, and then that one letter goes into a centralized service. And so I don't have to write 20 different letters, one for each of the different programs you're applying to. So anyway,
okay.
That's Michelle, was that your experience at UCSD? Did you apply to more than
Yeah,
one program?
so for UCSD, it depends on the departments where the program is. So if you're applying for programs in the same department, you can usually like double apply. And then for letters of rec, you generally just ask once for a letter of rec and then send all the emails to your professor or whoever's writing your letter, and they just submit the same letter for each one.
And then the other, thank you. And the other aspect of your question was, to be totally honest, trying to figure out how to word this politically nicely. Don't focus so much on the university name. Focus on the advisor whose lab you're going to be working in, right? And so they may not have a computational neuroscience PhD program, but look, if they have a computational neuroscientist doing interesting work that you like, then it doesn't matter. I don't want to say it doesn't matter, but like whether your PhD diploma says neurobiology or biology or neuroscience or data science or cognitive science doesn't matter as much as the research you do in the lab that you end up in, right? And so if there's somebody doing really cool work that you think is neat and you want to be in that environment, apply to work for them, this is what I tell students. If they want to work for me, I don't, apply to neuroscience, data science, computer science, cognitive science. I can take students from all of those PhD programs. If you want to work in the lab, figure out a way to get in, right? Like if you look at the people that have graduated with PhDs in my lab in cognitive science versus neuroscience, you can't tell who did which program. They all look very similar in terms of their research output, right? And employers don't care as much what that says. They, if you want to do academic research, it cares about the research that you do during your PhD, right? So, yeah. Now, when you get to the university, if you have the opportunity to take classes in more computational domains during your PhD, you should totally do that, right? So you get a neurobiology PhD, but ask if you can take a class in, math, do a, God help you, linear algebra for graduate students or, something like that. Try and take those classes to supplement your education, to build the degree that you want. But most of the PhD is not class taking. Maybe for the first 2 years, you'll take a couple of classes, and grad level classes are very different from undergrad classes. Undergrad classes, you study, take tests. Most grad classes are sitting around talking about papers that you had to read and critically engaging with those peer-reviewed papers. And sometimes, writing code, and, I'll make people in my graduate level classes. I teach teaching a grad class in neural oscillations or this kind of data analysis, neural data analysis in the spring, I want to say, a grad class. And so they have to write some code and stuff, but it's, as Michelle said in the chat, mostly journal clubbing, reading papers and then talking about what they did right, what they did wrong, how you might do things differently now, and so on.
Quick question. So, too loud. Is there something going on?
Nah, I like your voice synthesizer. That's awesome.
Oh my God, it does it every time. Second.
It's okay. Go ahead and ask. I'm enjoying it. You're.
Okay. So I'm someone who's doing mostly bioinformatics.
Okay, bioinformatics. Yeah.
Do you really? And this is too much. Let me know. I can turn off the voice modulator. But yeah, I'll tell it. 1 second.
It's okay. I've understood everything so far. It's not too much, but you're killing it. You said in 1 second, right?
Yeah. So
I speak DJ, man. I can hear you.
I'll stick with it, even though it says it's off on my software, so I have no idea what's happening. It is off now? Is it good?
You're good.
Okay, cool. That's happened like 4 times in classes, and I have no idea why, but I try to roll with it. Anyway, I study mostly bioinformatics at this point, and I kind of want to bridge this gap with kind of like neural computation, where I feel like a lot of kind of hardcore bioinformaticians don't want to talk about neuroscience in the way that like it's kind of akin to what you're talking about, where you kind of like redefine the paradigm of how you look at it. How would I try to kind of bridge that gap? Because I love the fundamental bioinformatics, but I also love just thinking about the brain in this way and trying to kind of just like shift how I'm working on what I'm working on now.
The reality is, if you want to do that, you got to play the very long, slow game, right? When I noticed these problems in the middle of my PhD, and that was in 2007. And as a lowly PhD student who hadn't published anything in a very kind of famous in my field lab, I can't just go and be like, hey, y'all, all doing it wrong. Nobody's going to listen to me. Like, get away, kid. Nobody cares, right? That's the reality. And so I kept the long game in my mind of like, okay, this is what I want to do. How do I get to that point in the long run? Right. And even honestly, in my undergrad, I had some of these ideas from having a conversation with my buddy. And they were, not very well formed because I didn't know the field well enough yet. But it wasn't like I'm going to go out and change the field tomorrow. Right. It's you gotta baby step it. In order to make any kind of change in the field, you first have to have credibility. Nobody's going to listen to you if you don't already have that credibility. And so I was like, okay, well, how do I gain credibility in academic research? Well, I should probably get a PhD, because otherwise nobody's going to listen to me. Like an autodidact, self-trained, whatever. It's real hard to make anybody listen to you, right? And also, I knew that there's a lot I didn't know, and so I was like, oh, PhD would be really fun. I have an opportunity to work on research problems that I find interesting, and you kind of play that game a little bit, and then eventually you get that credibility building up based upon actually doing projects that people find interesting, are well written. And then you can start to sort of push a little bit more at the field. And I know that's not like a fun answer, but I feel like building credibility will take you so much farther than anything else. Because credibility buys you room to then push back. And you're like, you can't say something is wrong until you're an expert at it already. You have to know how everybody does it inside and out already. And once you have that expertise, you can then say, here's where I go wrong. And if somebody pushes back and say, well, you don't know what you're talking about, you say, in fact, I do. Here's 3 papers I published on the topic. And here's the class that I teach on it. And here's the book I've written about it, right? Like, you build that credibility and then you can expand. But that's the reality of not just academics, but of everything, right? It's very difficult in any domain, business, arts and literature, law, whatever you go into, to just jump in immediately and try and like tear down the system, right? You have to build credibility to demonstrate that you understand how everything is being done right now, and then you can change it. I hope that helps.
Oh yeah, thank you. It doesn't matter if it's not a fun answer, as long as it's a good answer, right? I appreciate it. May
not be the only answer, but I think it's the most realistic path, right? There are certainly people out there, the Steve Jobs and Zuckerbergs for good and ill in the world, that like jump in immediately, right? And they build something or do something that has a huge impact on everything going forward. But that is, I don't know, luck, and it's not even like individual brilliance, right? It's like all of this confluence of things. And so it sucks, but the safer bet is go in and demonstrate the knowledge, and it doesn't hurt to know, right? You'll learn more anyway, and that's always useful.
All right, thank you.
Yeah, you're welcome.
And sorry for the voice modulator again.
Seriously, don't mind.
All right.
I can stick around for a little bit longer if anybody still has questions.
Feel free to unmute yourself or type in the chat if you have any questions.
Yeah. I mean, if not, I certainly don't mind. I'll go play catch with my kids. But
I actually had a quick question.
Yeah, what's up?
So I come from the world of tunicates and like Siona intestinalis. And with Siona, we have like an understanding of like cell to cell, like how the species develops. I was wondering if you've tried looking at like cell-cell interactions in combination with like neurological signal?
Cell-cell interactions, meaning like neuron to neuron.
Yeah, neuron to neuron.
Not directly in that. I've never actually done like a single unit recording myself, right? Like we'll take data that people have, where they have two neurons, for example, that they know are directly synaptically, monosynaptically connected, right? And we've played around with like analyzing data on those, but yeah, I personally haven't done it, right? Like we kind of see ourselves, in physics, which was as an undergrad kind of how I started learning. There's a separation between experimental physics and theoretical physics. Right? You have the people that like build the Large Hadron Collider and they collect these mountains and mountains of data, right? But it's testing theories, for example, like the Higgs boson discovery, right? That was theoretical physics. That was like, well, we think this particle should exist, right? That separation doesn't really exist in neuroscience. You can't, theoretical neuroscience does exist, but it's real hard to just be a pure theoretician. And so a lot of what we do is what you would call a theoretical neuroscience. We say, okay, given what we think the neurobiological genesis of these signals are these double exponentials of these postsynaptic currents, then there's only a certain number of ways in which mathematically these exponential currents can integrate to give us the signals that we're seeing. And we should be able to reverse infer from the aggregate signals that we're seeing the degree of excitation or inhibition that's contributing to those signals, right? So we can argue that theoretically, but in neuroscience, you can't just write a theory paper very easily, because reviewers will come back to you and say, well, yeah, but show it. And so we sit at this weird intersection of, we come up with these ideas, and we then have to try and figure out a way of showing it. And we'll just like reach out to a collaborator like Alison Motri's group or somebody that does animal work. And we're like, hey, we have this theory. You've got good data that would help us address this theory. Can we analyze the data? Would you be willing to share it with us? Right. And so we have to have some expertise also in the data analysis, but we don't have a lot of expertise in the collection. The only data that we actually collect ourselves in my lab directly are these intracranial recordings from the people undergoing brain surgery, but those are pretty limited. We might be able to get single neurons actually from them soon. It's technologically feasible, labs do it, and we are buying the equipment to do it. But outside of that, everything else we beg other people for data.
Yeah, that sounds like how a lot of bioinformatician lab works.
Yeah.
Thanks for your answer.
Yeah, you're welcome. Totally narcissistic field, right? Absolutely. We're narcissistic beings, right? Like nobody actually, probably nobody really cares about, how a nematode wiggles its tail, right? Like what we really want to know is like the fundamental neuroscience, because you want to understand how human brains work, right? That's why we use model organisms, because we hope that they can inform us something more broadly generalizable about how the brain works. And so you can try and fight and say, no, I'm not a narcissist, but we all are. Come on. Like we really do care about how our cells work. Actually, that said, I also care about how my dog's brain works, because she's awesome. Come on, come say hi. Come on. Any other questions? This is Ada, by the way. I think it's Ada Lovelace Day today too, isn't it? All right. Well, thank you, everybody. I'm going to drop my email in the chat because I actually, like I said, do tend to respond to emails. And so if you have questions about anything, I will do my best to either answer you or point you in the right direction. Thanks for having me.
Thank you, Professor Voytek, so much. Thank you all for everyone who attended.
Have a good quarter, yo. See y'all later.
Here's the person who hosted this. She can't. Her Wi-Fi
Oh,
is too
hello.
bad.
Yes, my Wi-Fi is really bad. Nice to meet you.
Nice to meet you. Bye.